Very much for the kind introduction. And also thank you for not only inviting me to talk, but putting on this great week-long workshop. And I originally titled this Challenges in Diffusion Tractography, but maybe a more appropriate title is Opportunities in Tractography, that I really think a lot of these opportunities are well suited for the Dipy team and Dipy software to solve some of these. So can you see full screen? Awesome. So like I said, challenges in diffusion tractography. As we've seen throughout many of the talks this week, the primary modality used to map the connections of the living, breathing human brain is diffusionmri fiber tractography. And Dipy, of course, provides us the tools to do this. It usually starts with some measures of the directions of greatest diffusivity that we think align with fiber, with the fiber orientations within each voxel. We can create these beautiful 3D vector fields that we step along in very smart ways. And we've heard smart ways to propagate what we call streamlines that you can think of as digital representations of groups of axons that start and end at the same point. We can do this throughout the whole brain, creating the human connectome, and then we can virtually dissect or study specific pathways and study them in health and disease. In this process of using tractography to study the brain as a network or as specific fiber pathways is proven valuable in studying normal anatomy, segmenting the brain, understanding development, disease, preoperative surgical planning. It, of course, makes very pretty pictures. So it's shown up as album artworks for pretty good rock bands and has almost certainly shown up in your social media feed. So there's clearly a tremendous number of neuroscience applications and a lot of attention paid to these techniques over the last 20 years. I like to ask, what's the problem? Well, because these make very pretty pictures and because these reconstructions are visibly believable, the results from these techniques are often accepted as the true connectivity in the brain. However, the actual process from acquiring the data to generating these 3D fiber pathways is a multi-step procedure with numerous assumptions and uncertainties that affect the ability of tractography to represent the true anatomical connectivity in the brain. So we need to take a step back and really understand the process of tractography and the challenges that we may face. To describe this or visualize this, I like to visualize the different steps in the tractography process and maybe variability or ambiguity or different decisions that we have to make with each step. We start with the acquisition that has uncertainties similar to any medical imaging, artifacts, motion, distortions, S&R effects, scanner effects, resolution effects. We have the local reconstruction step where we estimate the direction of fibers in each voxel. And like we heard from Max earlier in the week, this depends on which reconstruction method we choose, how complicated the fibers are in that voxel, the number of directions we sampled in, the B value or diffusion weighting, and maybe any pre-processing we've done up to this point. Then we have the tracking process itself that depends on how you start and how you stop streamlines and that logic that you use to move through that 3D vector field. And finally, we have what I call interpretation, which is really any decisions you make when you quantify structural connectivity. And all of these sources of variability or ambiguity ultimately obscure what we really care about right at the top here, which is single subject inference, determining the connections or maybe lack of connections of an individual, and population inference, which is determining how one population's structural connections may be different from another. So the theme of this whole talk is challenges associated with different steps. And like we've seen throughout the week, there's really excellent work that's tackling one or many of these steps at a time, many of which are implemented in Dipy. And I'm going to focus on just 3 kind of randomly selected throughout this entire pyramid of challenges. I'll talk about distortion correction, challenges associated with distortion. Earlier in the week, Dr. Bennett Lamman actually talked about this a little bit, so there's some overlap there. So I'll present the challenge and then a potential solution. Then I'll talk about a challenge called the bottleneck challenge, and I'll talk about some solutions to that. And then I kind of end with an open-ended challenge on what it means to define bundles or define fiber pathways of the brain. So starting with challenge 1, distortion. Diffusion MRI is usually collected using echoplanar imaging. And this suffers from geometric distortions that, of course, affect the geometry of the brain. It affects the signal, causing signal pileups or loss of signal. And relevant to this talk, it's been shown to affect not only the estimated fiber orientations, but the resulting fiber tractography bundles that we'd like to study using these techniques. So we, of course, have to correct distortions in our images. Distortion correction usually requires collecting more data in more time on the scanner. And this hasn't always been done in the past and isn't always feasible due to time constraints, or it may be a little more complicated on some scanners than others. So I'd like to use deep learning to enable susceptibility distortion correction with historical or limited data sets that don't include specific fancy sequences that allow us to do distortion correction currently. And on a more personal level, this is really my first step into deep learning, and really opened my eyes to what deep learning can and cannot offer us when it comes to medical imaging. At a high level, diffusion MRI is distorted in the phase encode direction because of the low acquisition bandwidth in this direction. And state of the art, or what I'm going to call state of the art, requires, in this case, reversing that phase encode direction, essentially creating equal and opposite distortions. You can plug this into your favorite algorithm. In this case, we like to use FSL's top-up algorithm that essentially estimates the middle ground and corrects distortion. So we ask, what if you just collected your regular old distorted diffusion data and didn't collect this fancy additional sequence? Well, our insight here was you usually collect some sort of structural reference image, T2 weighted, or in this case, a T1 weighted image that's not distorted. And we'd actually like to use this undistorted anatomical image to synthesize what the diffusion data should look like if it was also not distorted. So we're really synthesizing diffusion data sets with an infinite bandwidth. We're then going to plug the real distorted data and the synthesized data into TopUp, telling TopUp that this is an infinite bandwidth, and letting it correct our real distorted data. And we call this SynBO-Disco. We're synthesizing what's called a B0 image from the diffusion data set in Disco for distortion correction. And the deep learning we're using is actually very standard in computer vision. It's image to image or pix to pix with conditional adversarial networks. It's the same network that makes maps look like aerial photographs, day look like nighttime images. In our case, we're just going to ask it to make a T1 weighted image look like a diffusion image, but importantly, a diffusion image that is not distorted. And we've trained our data set using not only a lot of data, but different kinds of data with different distortion directions, different distortion magnitudes, different resolutions, TEs, TRs, different contrasts, and different qualities, good quality and bad quality data. So hopefully our network will generalize to unseen data if you were to use this at your own site or scanner. So what happens then is you collect your regular diffusion data, your T1 weighted image and your distorted data. You plug it into our pipeline and synthesize a perfect undistorted data set. You then plug the distorted and undistorted into TopUp and let TopUp correct your distorted data. And it works. Here we look at D distorted to U undistorted for a few different data sets with the T1 in the middle. In all cases, it visually corrects distortion. We can quantify this. Here we're using mutual information with T1 kind of as a proxy for how well we correct geometry. The distorted data sets have a low T1. If we were to use state-of-the-art processing, distortion has a low mutual information. If we were to use state-of-the-art processing, we improve the geometry of the images, and our algorithm here at the right improves the geometry almost as much as state-of-the-art. But it's important to point out that we're essentially using half as much data in doing this when this otherwise wasn't possible before. We can also quantify variability in some measures. State of the art, of course, reduces variability, and we reduced it almost as much. So our final images are geometrically more similar to T1. They closely match state of the art, and our quantitative indices are more reproducible. What's more exciting is this generalizes to all sorts of new data sets. Here again, D distorted to U undistorted. We grabbed about a dozen open source data sets and are correcting this without utilizing the reverse phase encode scans. And it seems that we're doing really well visually if you look at some of these data sets. So we've enabled this correction when it otherwise wasn't possible before, correcting geometry, correcting signal, reducing variation in modeling. We can apply this to old data sets or data sets with time constraints. And we're very excited that this is now actively being integrated into Dipy and the Dipy pre-processing pipeline. I'd really like to use this kind of idea for maybe correcting other contrasts that use EPI, or sort of a QA or quality control step in the entire diffusion processing pipeline. So I, of course, only talked about one of the many choices you make in the acquisition and pre-processing. And I wanted to at least point you to an ISMREM diffusion study group effort. There's actually many efforts coming to consensus on different diffusion MRI topics. This one happens to be being led by Jelle Verart. And the idea is to really understand and come up with guidelines, possible guidelines or standardization for image pre-processing. And the idea here is that there's really no consensus on the best pipeline, the best order of steps, or the best algorithm for any step or any artifact that generalizes to all data. So we'd like to understand where variability comes into play, which steps do or do not reduce variability, with the ultimate goal to make diffusion MRI a useful biomedical tool. And this of course covers all diffusion MRI aspects, of which tractography I think is an important part. And it's important to understand the impact of pre-processing on the tractography pipeline. So moving from acquisition to another challenge here called the bottleneck challenge. One thing we've seen throughout the validation literature for the last 10 years is this maybe pessimistic idea that the anatomical accuracy of tractography is inherently limited. And you see this over and over, these sensitivity specificity curves, where algorithms that do well at recovering the true anatomical connections very often have a high number of false positive connections. And those that are highly specific often don't recover all the true connections of the brain. And this seems to be true of different softwares, different streamlined propagation algorithms, different reconstruction algorithms, different resolutions, different species. And this challenge, or this sensitivity specificity trade-off, or this limited accuracy, has traditionally been attributed to two main challenges in the field. One, a very popular one, is this crossing fiber problem. And this is when two or more differently oriented fiber populations occur within the same imaging voxel. This causes a partial volume effect that can lead to ambiguous or incorrect orientation estimates, which of course affects the subsequent tractography. Crossing fibers have been shown to occur in a majority of voxels in the human brain, and we know that increasing or improving our resolution isn't going to solve this problem. And for the last decade, the crossing fiber problem has been cited as one of the major limitations of diffusion tractography. But the identification and characterization of this problem has led to a paradigm shift in the way we process data. Like we saw from Max's talk earlier, this crossing fiber problem led to dozens and dozens of different algorithms that are now capable of solving this problem, and has led to a development of methods to solve and overcome this problem. So rather than the crossing fiber problem, I'm going to focus on the bottleneck problem. And the idea of the bottleneck problem is rather than a local voxel level, this occurs at a global level or over many voxels. And the idea here is when multiple fiber populations converge towards the same narrow region, temporarily aligning and sharing the same orientation, before they re-emerge from this bottleneck region, current tractography algorithms can't choose the correct pathway when they re-emerge from this region. There's lots of possible pathways that can lead to the same orientation estimates. And this leads to a generation of a potentially large number of false positive pathways. And it really limits our ability to use tractography as a tool to explore potential connections and fiber pathways of the brain. It's been shown in simulated data, in human data, here's an excellent study of using tracers, histological tracers, and monkey data. So really, anytime two connections of the brain start and end at different locations, but at some point are within the same voxel and share an orientation, diffusion tractography faces a bottleneck. However, the prevalence or a thorough characterization of bottleneck regions hasn't been performed. So the aim of this work is to characterize the prevalence of bottleneck regions and identify where these occur in the human brain. And our idea is, just like the identification and characterization of the crossing fiber problem spurred development of all these algorithms, we hope characterizing the bottleneck problem will not only raise awareness, but also increase development of tools to overcome this challenge, or at least to quantify uncertainty in the tools that we develop. So to describe the bottleneck challenge, I need to introduce a little bit of nomenclature. Here we're going to characterize things happening within a voxel and within what's called a fixel, or a fiber bundle element, or you can really think of this as just an orientation or a peak within a voxel. So we're going to characterize voxels as single fiber populations. So here we have, this is just a cartoon of a couple bundles crossing, and we have 5 cartoon voxels here. But we characterize each voxel as a single fiber voxel or a crossing fiber voxel. So this is just traditional crossing fiber problem analysis. Then we're going to characterize each voxel as a single bundle voxel or a multi-bundle voxel by counting the number of bundles that pass through that voxel in any orientation. And finally, we're going to look at fixels or individual orientations and label each fixel as a single bundle or a multi-bundle fixel by counting the number of bundles associated with that orientation. And it's really these multi-bundle fixels, like you see in example C here, or example E here. It's these multi-bundle fixels in specifically large areas of spatially coherent multi-bundle fixels that represent bottleneck regions for tractography. So we perform bundle segmentation. We run 2 different bundle segmentation algorithms, TrackSeg and RICO bundles, like we've heard about throughout the week, resulting in 60 to 70 different white matter pathways. And then we assign these bundles to both fixels or individual orientations, and we assign those bundles to voxels. So this will let us count the number of bundles per fixel and per voxel. And it's important to point out here, or really, really overemphasize, that everything we're doing is specifically designed to underestimate the extent of this problem. We're only using these 60 or 70 very well-characterized, known anatomical pathways of the brain for which there's broad agreement on their existence and evidence through other species or tracers or dissection that these truly occur in the brain. And we're also using pretty strict thresholds on densities to assign these bundles to fixels and voxels. But once we've done this, we ask just 4 questions. What is the prevalence of multi-orientation voxels? So what's the prevalence of the crossing fiber problem? What is the prevalence of multi-bundle voxels? How often do bundles overlap? What's the prevalence of multi-bundle fixels or bottlenecks, and where do these occur? First, looking at the single and crossing fiber problem, we see there's a high prevalence of multi-orientation voxels throughout the brain. This agrees with previous literature. So here we're looking at a single subject and the average across a population, and confirm that most voxels contain more than one orientation, or most voxels contain crossing fibers. Next, looking at just the overlap of bundles and voxels, we find that many voxels contain multiple known bundles, with many voxels showing overlap of 7 or more different white matter bundles. And here we're just showing what's really just a bundle density for RICO bundles at top and tract seg on the bottom. And they show similarities of where these pathways overlap. So now is the interesting results, the bottleneck regions. So here we're showing individual voxels or individual orientations and counting the number of bundles associated with each voxel. Qualitatively, it's pretty obvious that most of these voxels are in this blue, orange, or hot colors. So we can already tell that nearly every voxel is going to be associated with more than one bundle and be a bottleneck. Quantifying this, we find that most voxels do contain multiple white matter bundles. Many are associated with 7 or more different bundles for both RICO bundles and tract seg. And where do these occur? So here we're looking at just a few different areas we decided to point out. First, the deep white matter of the occipital lobe oriented in the anterior posterior direction, covering actually a very wide dorsal ventral extent within the occipital lobe, as well as another bottleneck region in the internal capsule and traversing throughout the brainstem and midbrain, both of which contain, here we're showing 8 or more different white matter bundles, association, projection, and commissural all passing through these regions. And to really convince you that this is a problem or give you a better idea of the problem, here I'm extracting subsets of the bundles that pass through just a single voxel. And this may actually be a 2 by 2 grid of voxels. But we have very unique bundles that follow the trajectories of well-known bundles. And if we just pull out the centroid, we see that all of these bundles start and end at a unique point while traversing through the exact same voxel with the exact same orientation. So the idea is if you were to seed at any one of these points, the chances that you correctly go to the right ending location is very small. And this is why we have a huge number of potentially false positives in our tractography. So in summary, we've shown that a majority of voxels contain multiple orientations, a majority of voxels contain multiple bundles, and a majority of voxels within those voxels also contain multiple bundles. A unique kind of side finding that we had was that there's many voxels and voxels that are unassigned to bundles and are in these 2 specific atlases we used, which maybe represent areas of undescribed bundles that could be added to our atlases. And we'd of course like to use other atlases to better characterize this problem, like some of what we've heard of Lauren O'Donnell's atlases or the many great atlases that exist out there. Let me see. Oh yeah, so in summary, the main idea here is a majority of the voxels in the brain contain not only crossing fibers, but also bottleneck regions. So what does this mean? This is actually a best case scenario of the prevalence of this problem. It means that most streamlines are susceptible to this problem. In connectomics, we should expect a lot of false positive connections. When we study pathways or bundles, it means that absolutely is impossible to characterize or describe the existence of new pathways using diffusionmri alone, even if it's reproducible. Tractometry, or the characterization along and across bundles of maybe microstructural indices, certainly suffers from partial volume effects, where features, even within an orientation, are not specific to one bundle. And related to single fiber populations, these are relevant for microstructural modeling, where even a single fiber population, or what we traditionally consider a single fiber population, contains multiple pathways with potentially varying density, sizes, and distributions of axons. And this is also relevant for, like we heard earlier in the week, spherical deconvolution, where you need to estimate a response function. It means response functions aren't specific to one specific pathway. And there's varying literature on this as to whether this does or doesn't matter when it comes to reconstructing these estimates of fiber orientation. Good news, there's a lot of work overcoming and currently overcoming these challenges. Some examples are using non-local information or information across entire streamlines to filter streamlines or weight streamlines based on diffusion-derived properties, based on non-diffusion derived properties, myelin or T2 maps. Machine learning likely has a place in here to learn or integrate streamlined history or priors. And as the community is always doing, incorporating anatomical knowledge into this process is almost certainly helping. Whether that anatomical knowledge is knowledge of cortical folding or space or organization or the geometry, or like we heard about parallel transport or particle filtering tractography. And I think anatomical priors is really the key to overcome a lot of this bottleneck challenge. So related to anatomical priors, this is probably my favorite study I've ever done, and far and away my favorite title of any paper. Brain connections derived from tractography can be highly anatomically accurate if we know where white matter pathways start, where they end, and where they do not go. The idea of this entire study was we realized that most validation studies represented a worst case scenario for tractography. They were relatively unconstrained, didn't really utilize prior knowledge, weren't always done in humans. And we just wanted to know, do these get more accurate if we manually place regions, which is commonly done in real studies, real neuroanatomical studies of the brain, or using template-driven constraints? So we actually went through and used inclusion, exclusion regions, as you may do if you're manually dissecting these pathways, and showed that you can dramatically increase the sensitivity and specificity of tractography. So the funny thing here is, well, isn't this obvious if you can very clearly delineate where you want these things to go? The idea here, and the really important point of emphasis, is that current algorithms do in fact create subsets of streamlines that start at the right place, end at the right place, and follow the entire correct trajectory. So adding a little optimism to the diffusion tractography literature, anatomical constraints can dramatically improve our tractography process. And this was actually kind of reconfirmed in a community challenge this year led by Chiara Maffei and Anastasia Dipy from MGH, where just even very simple anatomical constraints improved accuracy. So this is great news. If we clearly define our bundles, we can have very anatomically accurate tractography. So let's look at the literature and see how they define these bundles that naively we'd think would have very, very clear descriptions of where they start, where they end, where they go, and where they do not go. But when it comes to some of these major pathways that have been studied for over 100 years, there is some variation in the literature in the naming and classification of these pathways. There's some debate about where they start, where they end, where their subdivisions are. So there's a diversity of definitions in the literature. Worse, how we define a pathway in words affects how we track them using our diffusionmri technologies, which may lead to differences in results and different scientific conclusions. So this becomes, like we saw, the acquisition and analysis. We think we have ways to solve that problem, but now we add on this anatomy, nomenclature, and taxonomy problem on top of the computer science problems that we've been talking about so far. So in this study, we'd like to understand how different white matter bundles are reconstructed, understand maybe if or when there's variability in their definitions. And this is really not a feasible goal anytime soon. And I wrote, come to a consensus on defining pathways, but really if we can maybe better understand how to define it, we can better understand how to more reproducibly track this. The good news is that variability in neuroimaging is not new at all. We decided to follow the lead of a consortium working on hippocampus segmentation, which is relevant for Alzheimer's disease. And here they recognize that you absolutely need to reduce variability in this process for meta-analysis or combining large data sets of results across labs. And they actually solve this problem, so we're going to do what they do. They first surveyed the field. They asked how different groups do this. They quantified the problem. They figured out where differences occur, came up with a consensus, and then evaluated their protocol. So we're going to do exactly this and start by surveying the field. So we invited participation from all sorts of labs. We have clinical sites, hospitals, neurosurgeons, tractography developers. In total, we ended up getting 42 different teams and over 130, 140 different collaborators. We gave them all the same 6 data sets. What we didn't tell them was it was really just 3 subjects scanned twice. And we actually gave them streamlines directly. So we eliminated all the sources of variation in that pyramid of variation that I showed earlier. We eliminated all variation associated with acquisition, reconstruction, and tractography. And all we asked was for 14 white matter pathways, define the pathway in words, and dissect the pathway and give us the results. In total, we received 57 different submissions from 42 groups, or 57 different protocols to dissect the same bundles. Visually, I'm going to flip through some results here. This is the cortical spinal tract. I like showing this because most people are somewhat familiar with where this pathway is and its importance. And these are the results across labs for how this is dissected for the same pathway on the same subject with the same data. And the main result here is there's some inter-protocol variability, and we can see differences in the connectivity and spatial extent of this pathway. Worse, if we go to a somewhat more ambiguous pathway, the superior longitudinal fasciculus, we get very different results. Some are one structure, 3 substructures, some include this arcuate fasciculus, some are more anterior, posterior, superior, inferior. The main result, again, is qualitative differences in the location, connectivity, and spatial extent of this pathway. And it's really important to emphasize that as of now, none of these are wrong. They've all been published or used in the literature to study the brain in some way. So we want to quantify this variability. And here we're showing a few different ways to quantify differences between bundles, cartoon bundles A and B and A and C. We can look at overall similarity in volume using the volume dice overlap. We can look at similarity of high and low density areas by just correlating the streamline density. We can look at average disagreement using a measure that we happened here to call bundle adjacency, which just asks when bundles disagree on average, how far apart do they disagree? And then we can look at where these bundles have streamlines in common and calculate a streamline dice overlap. So again, starting with the cortical spinal tract, here we ask where 25, 50, and 75% of groups agree that the cortical spinal tract exists. And the main result is reassuringly, more than 75% of groups agree on the general location and core of this white matter bundle, with maybe some small disagreements on like the thicknesses or lateral or medial terminations, or at the periphery of this pathway. When we look at streamlines, here we're asking where 25, 50, and 75% of groups agree a streamline is part of a bundle. The most amazing result here is that there's not a single streamline that 3/4 of the groups agree belongs to the cortical spinal tract. This ends up being true of all the bundles we studied. There's no agreement on individual streamlines, but general agreement on the volume or space occupied by this pathway. So here we show some of those measures I talked about, dice overlap, density correlation, and that disagreement measure across protocols, within protocols, and across subjects. The dice overlap is low to moderate. We get a slightly higher density correlation and a large disagreement. Reassuringly, using the same protocol on the same data results in a much higher dice, a much higher density correlation, and a lower disagreement. But the differences across subjects are on the order of differences across protocols. So overall, there's a pretty large inter-protocol variation. This is true of all the bundles we studied. Turns out the cortical spinal tract is one of the best case scenarios. The anterior and posterior commissures, the fornix, cingulum, arcuate fasciculus were generally much worse and much more disagreement. Overall, pathways have higher agreement in the densities, almost no agreement on individual streamlines. Things like the fractional anisotropy varied by as much as 10 to 15% across protocols. And the geometry can vary quite considerably. So we have a huge heterogeneity of white matter structures dissected when we use different dissection protocols. We propose this comes from a lack of consensus on how they're defined, on top of variability in integrating this definition into the tractography process. And my favorite example of this is the arcuate fasciculus. Quite literally 100% of the groups agree or wrote that it connects Wernicke's area to Broca's area, and its name means curved bundle. But where these areas are or how to unambiguously define them, there is very little agreement. There's disagreements on entire connections to lobules, disagreement on individual segments of this pathway, inclusion or synonymous or subcomponents of different systems. So very little disagreement even on the definitions themselves. Or sorry, very little agreement even on the definitions themselves. The good news is some pathways were better defined. The variability ultimately depends upon the scale which we're analyzing this. So maybe it matters if we're looking at individual streamlines or characterizing things along streamlines. Maybe it won't matter so much if we're measuring things based on the tract density or streamline density. And in the end, we propose that this is a combination of neuroscientists, neuroanatomists, and computer scientists problem. The anatomists need to explicitly define a bundle. Then we can use the technology we develop for tractography to recreate that definition. We can go full circle, ask the anatomist whether we met that definition and how we can better operationalize that definition to meet our tractography methods. And the good news is there's groups that are very much interested in looking into this. This first one from Daniel, who I believe is on the call. And this paper will forever be the anatomical reference that I give our students as they start out in anatomy. But along with this group, there's also groups really digging into nomenclature and taxonomy of the white matter in the human brain. And I really believe that given very explicit definitions, where they start, where they end, what they connect to, where they go and don't go, we can use already existing approaches, some of which we've heard this week, some of which query language. We may even develop very specific algorithms for individual bundles. I like this work using regions of interest in a very unique way for just the optic radiations alone. And along these lines, I'm interested in, as well as Joseph Yang from Royal Children's Hospital and Francois Roll in our group, are really interested in maybe starting to dig into this and really understanding how we define endpoints or connections, or how we think about positional definitions, whether things pass through or don't pass through or fan into other regions, and where our knowledge of these connections or disconnections come from, and what evidence we have that they really exist or don't exist beyond tractography alone. So I know the last one was open-ended, but I hope that by at least making you aware of the challenges or the different decisions, you can properly interpret these bundles that you're studying or these connectomes that you're studying. And I wanted to emphasize that rather than challenges, I really wish this had been called opportunities in tractography. I think there's many opportunities. Everything we've heard about this week from the denoising and patch to self to the parallel transport tractography and the propagation methods is really going to help us make tractography a useful biomedical tool. And so that's all I have. I'd like to thank not only the funding and my mentors, Adam Anderson, Bennett Lamb, John Gore, and Seth Smith, but also a lot of the students from the Massey Lab that have done a lot of the work that I showed today. So thank you very much.